Okay, welcome everybody to Chapter 7 in Gunstream, where we're going to discuss the structure of skeletal muscle tissue, and we'll also begin to address the naming and the function of various skeletal muscles. We'll also touch briefly on the structure of smooth and cardiac muscle. So we'll compare the structure and function of the three skeletal muscle or three muscle types: smooth, cardiac, and skeletal. We'll describe the structure of skeletal muscle and explain how it's attached to a bone or other muscle. We'll talk about the structure of a muscle fiber, talk about what a motor unit is, and talk about the role of the neuromuscular junction and where they're found. We'll describe the physiology of muscle contraction, and we'll explain the cause of and recovery from oxygen debt, and we'll also explain how the all or none contraction of muscle fibers allows graded contraction in whole muscle, and we'll explain the relationship between a muscle's origin, insertion, and action. We'll talk about some of the major skeletal muscles, we'll name them, and we'll give a job description. By providing you the origin, the insertion, and the action, you'll have a better idea of the function of each of the major skeletal muscles we'll cover. We'll identify these on a diagram, and then we'll end by talking about disorders of the muscular system, both infectious and trauma-induced. So muscle tissue is special in the body because it's the only type of tissue that can generate force as a result of contraction. This moves the body both internally and externally. Cardiac and smooth muscle are primarily responsible for internal movement, and they are not under our voluntary control, while skeletal muscle provides voluntary movement and is under our conscious control. So the three types are skeletal, smooth, and cardiac. They all have the ability to contract, but again, they're found in different places in the body, and of the three, only skeletal muscle is controlled by our conscious will. Skeletal muscle is composed mainly of muscle fibers that extend the length of the muscle, arranged in fascicles with connective tissue surrounding successively smaller groups of muscle tissue. The connective tissue is there to hold the muscle together while it contracts. In addition, it also translates the force that the muscle produces to the structures to which it's attached. So connective tissue, very important in muscle function. It surrounds each fiber, each fascicle, and also at its most superficial, the muscle itself. It produces tendons that attach muscle to bone, and it establishes broad, flat layers of connective tissue called aponeuroses to attach muscle to other muscle and to connective tissue. And we'll see a couple of examples of aponeuroses in our skeletal muscle diagram. One that runs across the surface of the cranium, which attaches the frontalis muscle, your forehead, to the occipitalis muscle, which is on the rear of the skull. And then we'll also find another aponeurosis at abdominal pelvic midline, where many of the abdominal pelvic muscles insert at midline in order to generate force. So what you're looking at here is a diagram of what skeletal muscle looks like. These are unusual cells in that they are multinucleate. When skeletal muscle is formed, what happens is that the precursor cells, which in the embryo are the mesoderm, that are going to become muscle cells, actually gather together in clusters, and they lose their intervening cell membranes to generate these long cylindrical multinucleate cells that produce massive amounts of muscle protein for the purpose of contraction. This is unusual in the body. There's only a few examples of multinucleate cells in the body. Skeletal muscle is one example. The cells that produce the platelets, the megakaryoblast, are another. We find those in red bone marrow. And we also find them in human development when they form something called the syncytial trophoblast. But generally speaking, when we look at a human cell, it has a single nucleus bound by a single cell membrane. But this is the exception to the rule. The sarcolemma is the plasma membrane of the muscle cell. It's quite extensive because it runs the entire length and width of the muscle cell. The sarcoplasm is what we would term the cytoplasm of the muscle cell. So the prefix sarco, as you probably guessed, means of or pertaining to muscle. The myofibrils are the contractile elements. They contain actin and myosin, which interdigitate inside the muscle cell and use ATP as an energy source. And the way that contraction is produced is that the thick and thin filaments slide over the top of each other in a ratchet-like mechanism using ATP for power. And we'll see exactly how that happens in just a second. Each myofibril consists of repeating contractile units called sarcomeres. We're now actually inside a single muscle cell here, and you can see that there are stacks and stacks of these myofibrils occupying the bulk of the sarcoplasm of the muscle cell, and that they're arranged in alternating overlapping fiber units. Okay, the area that runs from what we call the Z disc to the Z disc is defined as a single sarcomere, but you can see that within the sarcomere are the thin actin filaments and the thick myofilaments, and they're what actually going to produce the force at the end of the day. The sarcomere runs from Z line to Z line. The actin filaments are attached at the Z line and they extend towards the center of the sarcomere but do not actually touch. Okay, so you can see here as an example, this is a Z line here, this is a Z line here. The blue areas are the actin filaments, and the red are the myosin filaments, which is we're going to find out are attached to the middle of the H zone. Okay, so the myosin essentially to produce the force reaches up and attaches itself to the actin and then pulls on the actin, drawing the Z discs closer together. That shortens the sarcomere, which in turn shortens the myofibril, which shortens the myofiber, the muscle cell, and that is going to in turn shorten the entire muscle. And that's where the force comes from. What you're looking at here below where we see the myofibril pulled out of the muscle cell is essentially a two-dimensional look at the alternating bands that run throughout a single myofiber. You can see the I band, the A band, the Z line, and then within the A band, you can pick up the M line, which is what the myosin filaments are attached to, basically permanently. It's like their anchor point. And then around them is an area where there's no overlap of actin and myosin, and we call that the H zone. Okay. What varies in length is the degree of overlap when the muscles actually contract. So what will happen as the muscle fiber shortens is that the actin and the myosin filaments will overlap more extensively. The result is that the H zone will shrink, and also the I band will shrink as the overlap becomes more extensive. You can see here in its pre-contracting state the microscopic view of myosin and actin. Myosin is the thick filament. It has these golf club shaped heads which are capable of binding both ATP and also attaching to actin. So they are really where the force is produced in muscle contraction. The actin is an unusual molecule. It's arranged, attached to these strands of what we call tropomyosin, and the actin binding sites that would interact with the myosin are blocked by a molecule called troponin. And so what happens when muscle contraction initiates, as we're going to see in a minute, is that the troponin has to get out of the way of the actin and the myosin in order for cross-bridging to occur. Until that happens, no cross-bridging is possible. And we're going to find out that the trigger for this is the release of calcium inside the muscle fiber, which leads ultimately to the contraction. So the myosin filaments reside within the A band and do not come into contact with the Z lines. They don't reach all the way to the Z disc. They never do. Here you can see a sort of a three-dimensional look at the inside of a single muscle cell, noting that you've got this extensive network of membranous sacs, which are essentially the muscle cell's smooth endoplasmic reticulum, contains large amounts of calcium, and upon stimulation by nerve fibers, what happens is that the calcium stored inside these sacs is released into the sarcoplasm of the muscle tissue, and that's what's going to attach to the troponin and ultimately result now in actin-myosin cross-bridging, which is what provides the power for contraction. Along the muscle cell is the sarcoplasmic, or I'm sorry, the sarcolemma running along the entire cylindrical surface of the muscle cell, and there are occasional invaginations of the sarcolemma which dip deep down inside the muscle cell called T-tubules. So this is provided a way for the electrical signal that radiates across the muscle cell membrane to get into the core of the muscle cell where it triggers the calcium release. So you could think of this in a sense, the T-tubules, as a type of wiring that allows the electrical signal that releases the calcium to reach every square micrometer of the muscle cell. They come into very close contact with these terminal cisternae, these bulges at the edges of the sarcoplasmic reticulum where the bulk of the calcium is actually stored. So the electrical signal that radiates down the T-tubules can actually jump across to the sarcoplasmic reticulum, and that's what will ultimately trigger plasma proteins that will release the calcium into the sarcoplasm. So this whole webbing or network of sarcoplasmic reticulum runs around and between every myofibril. The neuromuscular junction is the point where a motor neuron axon terminal stops just short of actually touching the muscle cell membrane. Across this tiny gap is going to be secreted a neurotransmitter, generally acetylcholine, which is what's going to induce the change in electrical state in the muscle cell membrane that will ultimately radiate longitudinally and down the T-tubules that will produce the calcium release. Each muscle fiber is innervated and controlled by a motor neuron, and without this stimulation, the fiber isn't capable of contracting. So anything that interferes with the neuromuscular junction function is going to interfere ultimately with contraction. It will produce a state that we know as paralysis. If we couldn't, say, issue the neurotransmitter, or if the nerve that innervated the muscle were damaged somehow so that the message to release the neurotransmitter never got sent, the muscle, as a result, would have no command to contract, and the result would be no movement. Motor units are essentially defined as a motor neuron and all the muscle fibers that it comes into contact with. This is under very precise control. There's a motor unit with very few muscle fibers for muscles that have to perform very delicate, finely controlled movement. In larger muscles where gross movements are all that's required, motor units can come into contact with hundreds of muscle fibers at one time in order to generate movement. So, for instance, the muscles in your fingers and your hands would have this almost one-to-one correspondence between neuron and muscle fiber, whereas in bigger muscles such as your glutes, the muscles of your upper limbs, you're going to have this less precise interaction where you have a single motor neuron talking to hundreds or even thousands of muscle fibers at the same time. Motor neuron activation causes contraction of the associated muscle fibers. We're going to find out that the difference in contractile force is generated by a couple of things. One is increased recruitment of motor units. The more motor units we recruit for an activity, the more power we can produce. And we're also going to find out that the more frequently the motor neuron fires, the more power we're going to generate within a single muscle fiber. So the neuromuscular junction is that gap between the axon of the motor neuron and the sarcolemma of the muscle fiber. The space is defined as the synaptic cleft. The axon tip contains neurotransmitter, and most of the time that neurotransmitter is acetylcholine. The nerve impulse as it travels to the end of the axon causes calcium to move into the axon tip, and that results in vesicles containing acetylcholine fusing with the membrane that surrounds the axon terminus, and that releases the acetylcholine into the synaptic cleft. What happens next is it'll work its way across this tiny physical gap. This is actually the slowest part of muscle contraction. And then it'll bind to acetylcholine receptors, which will then open up ion gates, resulting initially in a sodium influx that will lead to the production of an action potential, a type of electrical signal that results in a depolarization longitudinally and through the T-tubules all along the length and down the width of the muscle fiber that leads ultimately to calcium release inside the muscle cell and contraction. And so you can see what it looks like here in diagram. This is the motor neuron. You can see the axon termini, and these are the neuromuscular junctions above which the axon terminals sit. The receptors on the sarcolemma are just beneath each of these terminal areas, and they're what's going to respond to acetylcholine release. So that's how the electrical signal moves from one cell type to another. It's very similar to the way nerve signals are propagated within nervous tissue as well. It's the use of neurotransmitters that allows us to hop over that gap. We should note that in any electrical activity in the body, this is the slowest part. The time during which the neurotransmitter is released and the time that it takes for it to bind to its receptor on the target cell membrane is much, much slower than the movement of the electrical signal from the cell body to the axon and ultimately to the axon terminal. So when you look at things like neuromuscular junctions, or when you look at synapses in the nervous system, the fewer of those you have, generally the more rapidly firing or the more rapidly responding that electrical pathway will be. The more of these you have, the slower it will be. Muscle contraction involves a lot of rapid structural and chemical changes in a muscle fiber, explained by something called the sliding filament model. Essentially, you can see here the mechanism of contraction. Acetylcholine released by the axon terminus binds to receptors on the sarcolemma that causes release of calcium inside the muscle cell from the sarcoplasmic reticulum. The calcium binds to troponin on the actin filaments, and that causes the actin filaments to pivot, and that exposes the active sites so that actin-myosin cross-bridging can begin. As long as there's calcium in the area and there's ATP to provide energy for contraction, contraction will continue. The myosin cross-bridges attach to the actin at their active sites, utilizing ATP for cross-bridging. This causes them to bend and pull the actin filaments towards the center of the sarcomere. The cross-bridges detach and reattach at successive active sites, kind of like a ratchet mechanism. Imagine you jacking up your car after you have a flat tire. Okay, the handle of the jack and the actual jack itself, those little interdigitating teeth, they're very similar to the action of the myosin heads with the actin binding sites as we grab and pull and grab and pull and slowly move the Z discs closer and closer together within a single sarcomere, and as a result shorten the entire myofibril and muscle fiber in a larger sense, producing the force of contraction. The cross bridges detach and reattach to the active sites as shown in steps 5 and 6. Okay, so you can see those steps here. The cycle repeats as long as there's calcium and ATP around to drive this interaction. Acetylcholinesterase, which is an enzyme that breaks down acetylcholine, is released from the sarcolemma and causes a decomposition of the neurotransmitter that bound to the receptor on the sarcolemma to begin with. This is what ultimately is going to help end the change in membrane potential that resulted in the contraction in the first place. This prevents continual stimulation of the muscle fiber and allows it to get ready for the next stimulus to come. So very important that we understand that it's only while the calcium is around and the ATP is in ample supply that the contraction can continue. When we reverse this entire process by releasing the acetylcholine from its receptor, what happens now is that the calcium that's in the sarcoplasm ends up getting pumped back into the sarcoplasmic reticulum. As a result, it detaches from the troponin, which was critical in pivoting the actin so that the myosin could interact with it. What happens then is that the troponin will then move to block the interaction between actin and myosin, and at that point no cross bridging is possible and the muscle goes back to its resting length. So you can see here what this looks like in 2 dimensions. Again, notice here that the A band does not change in thickness. The distance between the Z lines decreases. The I bands and the H zone also shrink as well. The I bands being these areas here and here, and the H zone being the hypochromatic zone that represents the area of lack of overlap between myosin and actin at midline of the sarcomere. So acetylcholine binds to receptors on the sarcolemma. It stimulates the release of calcium from the sarcoplasmic reticulum. The calcium then binds to the actin filaments at troponin, causing the active sites to be revealed so that actin and myosin cross-bridging can occur. Myosin cross-bridges attack to the actin active sites using ATP for energy. This causes cross-bridge formation and release, and this causes the actin to be pulled towards midline of the sarcomere. The result is contractile force. The cross bridges detach and reattach to the active site, and the cycle repeats again as long as calcium and ATP are around, and we've already detailed the role of acetylcholinesterase. We should also point out that in addition to acetylcholinesterase breaking down neurotransmitter in the neuromuscular junction, diffusion also plays a role. Diffusion, of course, is the movement of substances down their concentration gradient, and what happens over time is that the bound neurotransmitter eventually diffuses away from its receptor simply as a result of concentration concerns in addition to enzymatic breakdown. If this didn't happen, we would continuously stimulate the muscle until the ATP was gone, and at that point we would enter a condition known as rigor. Now, endurance training increases the efficiency of aerobic cellular respiration by increasing the number of mitochondria, which generate most of the ATP in living cells. It also increases the efficiency of obtaining oxygen from the blood. This is because there's a pigment inside muscle tissue called myoglobin that serves as an oxygen storehouse, and the more endurance training we have, the more myoglobin we generate in the blood, the more oxygen store we have. The result is that we're less likely to exhaust ourselves even when the ability of blood to bring oxygen to the tissue is diminished. Fatigue is defined as the reduced ability to perform work, and there's 3 kinds. There's synaptic fatigue, muscular fatigue, and fatigue that occurs at the neuromuscular junction. This occurs with continuous nervous stimulation. We see a decrease in contraction ending with an inability to contract when we're stimulated. Causes include the accumulation of lactic acid and carbon dioxide as we become unable to deliver oxygen to the muscle quick enough to feed the energy debt. A depletion of ATP is a major player as well. Heat from muscle contraction is used to maintain normal body temperature in the act of shivering and also in normal contraction. In fact, every chemical reaction in the body releases a small amount of heat and is responsible for maintaining our body temperature around 98.6. Heat production in muscle is caused by cellular respiration and other chemical reactions within the cell as well. Contraction of a single muscle fiber is going to be relative to the electrical signal that is designed to produce the contraction. The threshold stimulus is the minimal stimulus that will cause a muscle fiber to contract, and when it does so, it always exhibits what we call an all or none response at the level of the muscle fiber. So either the muscle fiber will contract or it will not. Contraction, though, is not always proportional to the stimulus strength, and this is because of this all or none phenomena. Again, the difference in power produced by muscle is as a result of increased frequency of firing of the neuron at the neuromuscular junction and also the increased recruitment of motor units. Muscle contractions can be recorded as myograms. A single contraction due to a threshold stimulus has three distinct intervals: the latent period, the period of contraction, and the period of relaxation. You can see those different periods here. The latent period is where we're preparing chemically to generate a muscle contraction. The force is generated in this rise to peak, and then in relaxation what happens is that we stop the actin-myosin cross-bridging and slowly the muscle fiber goes back to its resting length. Again, this is done at the level of a whole muscle. So what you're seeing here is really the summarized action of many myofibers, okay, not an individual muscle cell. And this is one of the reasons why we're going to see changes in the amount of force produced as we change the frequency of the nerve signal to the muscle, and also as we change the number of motor units that we recruit. So if the muscle is stimulated after relaxation, we generate this familiar peak and drop, okay? And the reason for that is we allow now complete relaxation between induction of contraction. If the muscle is stimulated prior to fully relaxing, we have what we call summation, where the power produced in each successive electrical command is greater than the power of the previous electrical command. And the reason for this is that due to the fact that the myofibrils have not completely gone back to their resting length, you still have some degree of overlap and cross-bridging occurring between the actin and the myosin. And then when that second or that subsequent nerve stimulus comes in, we're able to generate, as a result, more power. The force of the individual twitches combines, and this increases the force of the contractions. And again, that's because we haven't gone completely back to our resting length. And so what you're seeing here again is that the higher the frequency of these electrical signals, the more power we produce more quickly. Okay, this is a stair-step effect, which we call TREPI. Okay. If the stimuli are so frequent that relaxation doesn't happen, then what we generate is something called tetanic contractions, and you can see those in the bottom trace here. This is a tetanic contraction. You don't see any drop in force. Now, you can't sustain this for a very long period of time because the muscle will rapidly run out of available oxygen, available ATP, and we'll also have a buildup of lactic acid that's going to eventually interfere with the function of the sarcomere. Okay, so this is something that generally doesn't happen very often. Generally, the way that we produce tetanic contractions is not by jamming every muscle fiber on full blast at the same time. What we generally do, even when we think we're producing maximal force, is that the nervous system is subconsciously alternating the motor units that it stimulates in order to give certain motor units a break, a chemical break, so that they can continue now to generate contraction the next time they're stimulated. Full, complete tetanic contraction with every single muscle fiber producing maximal force would only last a few seconds. Tetanic contractions lack even partial relaxation on the myogram, and this is due to a rapid series of stimuli. This is the usual way that muscles contract to produce body movement, but again, remember that what looks like no resting period for the muscle on the myogram actually contains rotating periods of rest for varying motor units on an alternating basis. So very important to understand that. Graded responses are varying degrees of contraction in whole muscle due to the presence of different motor units responding to different thresholds of stimulation. We call this recruitment. Maximal stimulus activates all motor units and generates maximal contraction. Further increases in stimulus do not produce a stronger contraction because it's not possible to recruit any further motor units up and above the maximum number. Even when muscle is quote-unquote relaxing, there's a state of partial contraction called muscle tone that works under our conscious effort to keep the muscle ready to respond. This is due to alternating activation of different motor units even when we're at rest. Loss of nervous innervation results in loss of muscle tone and eventually atrophy. Bottom line, if you don't use a muscle, you'll eventually lose function in that muscle because biologically it doesn't make sense to give energy and resources to a structure that's not being utilized. So what we're looking at here is a typical skeletal muscle. This is the biceps brachii. It's the big muscle in your upper arm. It produces flexion at the elbow when it's stimulated. And there are several important features that we need in order to describe this muscle's location and action. One is the insertion, which is its attachment to the movable bone in the joint. In this case, we see attachment down here to the radius. Okay, you can see here the bone on the thumb side of the forearm contains an attachment point for the biceps brachii. The origin is the immovable muscle attachment. In that case, we're looking at the attachment to the scapula. And isotonic contractions are defined as those that produce movement at a joint, while isometric contractions do not produce movement, but they both generate force. It's just that you don't see the movement. An example of an isometric contraction would be if you were to push on a wall, okay? You would be generating force, you would be burning chemical energy, and you would be strengthening the muscle, but you wouldn't be producing any movement. While an isotonic contraction would be something like a push-up, okay, where you're elevating the body up and down off the floor. Strength training causes hypertrophy of skeletal muscle due to an increase in the amount of muscle protein that the muscles build. Endurance training improves the efficiency of muscle action, but it doesn't generally result in hypertrophy. Instead, we increase the number of mitochondria and blood vessels to the muscle so that it's less likely to fatigue. This increases the amount of oxygen, nutrient, and ATP supplied to the muscle. The reason this is an important thing to understand is because depending on how you train, your muscles will develop differently. If you look at the legs of sprinters, they tend to be quite big and powerful, but easily fatigued, whereas if you look at the legs of a marathon runner, they tend to be long and stringy, but they tend to fatigue very slowly, if at all. and that's because of the difference in training regimen that generates in the sprinter a large number of myofibrils, but not necessarily a concomitant increase in mitochondria and vascularization, whereas in endurance training, we see an increase in vascularization and mitochondria, and also amounts of myoglobin, and we don't necessarily see a concomitant increase in muscle bulk. Okay, muscles function in groups that are arranged to provide opposing movements. Agonists generate an action while antagonists oppose the action. Agonists and antagonists contract alternately, and this is in order to produce fluidity of movement. If we didn't do this, our movements would be jerky and difficult to control at best. A perfect example of an antagonistic muscle pair is the biceps brachii in the upper arm, which flexes the elbow, and the triceps brachii, which will extend the elbow. And they oppose each other during elbow movement. The result is the ability to bend the elbow in a fluid fashion. If the bicep brachii didn't have any antagonist opposing its movement, when you bent the elbow, it would simply flex shut very, very rapidly. Same thing would be true if the tricep didn't have any antagonistic muscle group. It would extend very rapidly. And so when you look at joints, you generally see the prime mover on one side of the joint along with any muscles that assist that movement. We call those agonists. And the antagonists tend to be on the opposite side of the joint and generate the opposing movement.